Dear colleagues, my name is Ondřej Lerch and I am a scientific director in International Neurodegenerative Diseases Research Center. It is my pleasure to discuss here, on behalf of INDRC Scientific Advisory Board, the potential ways for amplifying efficiency and accuracy in dementia drug development. Next slide. Chronic and progressive brain disorders of aging, such as Alzheimer's disease and other neurodegenerative disorders, are a major source of prolonged disabilities that have both devastating emotional and economic impacts on patients, their caregivers, and families. Worldwide, the number of affected people, disease duration, and the cost of care are spiraling out of control. Costs associated with AD are estimated at $706 billion a year in the US alone. This figure is expected to rise sharply as population ages. The World Health Organization recognizes dementia prevention as a public health priority. It aims to increase awareness and create programs to reduce the burden on individuals living with the disease, their families, and caregivers. Early diagnosis and preventative interventions may help to delay the symptoms onset and smooth down progression. However, the overall disease burden will continue to rise unless effective pharmacological treatment options are available. Next slide. Despite intensive research in the last several decades, we are still searching for efficacious treatment to slow disease progression. Over the last decade, over 95 phase 2 and 3 AD clinical trials have been unsuccessful. The total cost of these trials was estimated to approximately $42.5 billion US. The most significant costs, of up to 57%, were incurred during phase 3 of clinical development. AD drug development faces a set of unique challenges. The average drug development costs in dementia field is $5.6 billion. The process takes approximately 13 years from preclinical studies to the approval by the FDA. This compares unfavorably with an estimated cost of cancer treatment development of $793.6 million per agent, or $2.8 billion per agent among all other therapeutic areas in the pharmaceutical industry. Further, there are multiple reasons why many new dementia interventions fail to gain market access. These include the long preclinical stage and progressive nature of the disease, inadequate drug dosing or penetration through blood-brain barrier, insufficient target engagement, inadequate stratification and trial enrollment, suboptimal trial design, the choice of suboptimal clinical outcome measures, long trial duration, increasing the risk of losing patients, the need for extensive enrollment involving multiple sites and tightly coordinated project management, or erroneous treatment targets. Despite this, 3 monoclonal antibodies against amyloid beta have recently succeeded in gaining FDA approval. These are aducanumab, lecanemab, and donanemab. Despite this, significant unmet needs remains to be addressed. Primarily, the mitigation of advantage side effects, especially amyloid-related imaging abnormalities, or ARIA. Other concerns include long-term effectiveness, as well as beneficial effects to broader and more diverse group of patient populations. Next slide. The multifactorial nature of Alzheimer's disease and related dementia presents significant challenges, particularly in capturing the complexity and heterogeneity, selective neuronal vulnerability, and specificity of affected brain regions. The use of the word multifactorial is often a rhetorical concept, context-specific and variably related to complex or lots of things. Neurobiologically, the multifactorial concept may describe how Alzheimer's disease and related dementias impact different molecular components, cells, and/or brain regions with certain hierarchical levels exhibiting differential susceptibility to degeneration. Meanwhile, the balance and timing between risk, modifiable or non-modifiable, protective or resilience factors across the lifespan that define the delicate boundaries between biologically defined and clinically defined disease is poorly understood. Clinically, the concept may translate to describe diverse symptom presentations, risk factors, and patient progression patterns. On a social and public policy level, the concept may characterize the complexity of creating effective public health strategies or equitable care practices for a disease that manifests differently across population and environments. Previous drug trials may have had multiple limitations. Various strategies have been employed to address these, such as increasing research volunteer sample sizes and extending follow-up periods to improve the internal and external validity of the results. Additionally, study sample stratification based on genetic and phenotypic characteristics of risk and resilience factors has become a key design feature to enhance data accuracy further. Yet, integrating modern approaches such as proteomics and transcriptomics holds the promise of refining RCT design and conduct by offering deeper insights into disease mechanisms and treatment effects at molecular level. Next slide. Proteomics and transcriptomics are emerging as transformative tools in developing clinical intervention for Alzheimer's disease and related dementias. These advanced methodologies allow for comprehensive analysis of proteins and gene expression profiles, providing far more precise, cost-effective, and efficient insights than traditional approaches. Though initially employed in basic research, proteomics and transcriptomics are now finding applications in clinical settings, enhancing the capacity to design personalized treatments. By examining the entire protein or RNA landscapes of patients, these techniques offer highly accurate data and predictive insights for individual patient profiles. Adapting proteomics and transcriptomics in Alzheimer's disease drug development faces significant challenges, primarily from data sparsity and insufficient sample sizes. The complexity of these disorders requires extensive, well-characterized datasets to identify and validate potential biomarkers and therapeutic targets accurately. However, obtaining sufficiently powered genetic data is difficult, particularly when focusing on specific disease manifestations. Additionally, the lack of diversity in available dataset further complicates the development of effective treatments, as most studies have historically relied on European ancestry data. Such information is unlikely to apply globally because the forecasted most significant dementia burden is mainly projected in nations not of European origin. Also, there is a critical need for standardized methods to ensure data harmonization and interoperability, which is essential for collaborative research efforts and maximizing the utility of available data. Addressing these challenges is vital for advancing precision medicine approaches in Alzheimer's disease and related dementias and improving the success rates of new drug development. Next slide. Advanced computational technologies may present transformative and unknown opportunities for advancing analytical methods in proteomics and transcriptomics, in Alzheimer's disease and related dementia drug discovery that should be further explored. High-performance computing maximizes computational capacities to tackle complex, large-scale problems across basic science, clinical research, and industry. Quantum computing leverages the principles of quantum mechanics that could be used to analyze and model rapidly the complex interactions of proteins and gene expressions, and aid in identifying biomarkers and predicting biological pathways. artificial intelligence and machine learning driven analytics could analyze large-scale proteomics and transcriptomics dataset, identifying patterns and predicting disease-related biomarkers. Additionally, model-based approaches representing ATN frameworks' pathophysiological mechanisms may contribute to this effort. Next slide. If you are interested in more detailed discussion, INDRC has already published the editorial on this topic in the Journal of Prevention of Alzheimer's Disease in October 2024 that is available online. Next slide. Finally, INDRC, along with Frontiers, Vitality Medicine and Engineering, Campaign to Prevent Alzheimer's Disease, Brainwatch Coalition, and CLARA have formed several working groups to advance this topic for both academic research and application of advancements in real-world healthcare settings. Major objectives of these work groups are to identify and validate novel diagnostic and therapeutic technologies, develop frameworks for regulatory approval and clinical adoption, survey and review methodologies for proteomic and transcriptomic integration, create collaborative networks of researchers in Alzheimer's disease and related dementias. Further, to facilitate data harmonization and interoperability standards, encourage non-competitive partnership with pharma and biotech, to propose ways to utilize advanced computational methods such as AI or quantum computing to enhance biomarker discovery, to create collaborative effort to improve data sharing and standardization, including researchers, clinicians, and industry leaders, and to engage in multi-institutional efforts to integrate proteomics and transcriptomic data sharing and analytic approaches. Next slide. First webinar on this topic has already been held on March 27th, 2025. And if you are interested in further collaboration, please don't hesitate to reach out. Next slide. This work is on behalf of INDRC Scientific Advisory Board and has been supported by CLARA grant Horizons Europe. Thank you for your attention.